It's Jim, the author behind the blog, Reverse Engineering the Origins of SARS-CoV-2. This is going to be a vlog about the Leblik narrative versus mine, and their evidence versus mine, and what I can poke holes in on their side. So this is going to be about Matt Ridley, and he was invited by Jay Bhattacharya to appear in front of an NIH group in Bethesda, Maryland, to discuss the lab leak debate, which is a step in the right direction. Don't get me wrong, I'm a lab leaker myself, but sometimes those that claim to have the answers are the ones that are part of the cover-up. So that's part of my insider's narrative here, which a lot of people are interested in, thank God, but some of you are. So when it comes to a cover-up, you have to kind of be careful about who's doing it. A lot of times you think the CIA, the CCP, the PLA, Bat Lady Shi, Christian Anderson, Bob Gary, other virologists, natural origin virologists, even Fauci or Baric, to a certain degree. No, I will contend to someone like Matt Ridley. He is the one covering up a lab leak because we have more than enough evidence to point at the culprits. It's just not the ones he wants to point at for various reasons I'm not going to try to speculate on. So here it's Jay inviting Matt to come up on stage in the NIH. This happened, I don't know, we're recording this in beginning of April 2026 as it is already. So this was like late March.
This series then is a step toward fostering that kind of dialogue. And in that spirit, I'm absolutely delighted to welcome today's speaker, Lord Matt Ridley.
Lord Matt
science
Ridley.
Writer whose work spans evolution, genetics, and the origins of human society. He was educated at Oxford University, where he conducted scientific research on evolution before starting his career as a science journalist at The Economist, serving as a science editor, Washington correspondent, and American editor.
He has the credentials. That's why he's invited on stage. That's kind of what this lab leak debate's turned into. It's about credentials, apparently, not about evidence.
Understanding human behavior, culture, and innovation. His books, which include The Red Queen. Anyone else read The Red Queen by him? I absolutely love that book. If you haven't read it, please do.
So here's Jay pimping The Red Queen. I have not read it, but even the natural origin scientists have like him for that. That is, he considers himself the modern day Darwin because he studied birds. That's part of the reason why he doesn't understand this lab leak debate, because he never studied bats. I read The Rational Optimist. That's how I knew Matt Ridley pre-pandemic. So I basically knew of him, and I liked him, and I respected him. So I was glad to see him as part of the lab leak narrative, the lab leak debate. But after DARPA Diffuse leak with Barrick's name on it, I was like, wow, this guy is going in the wrong direction, and I'm going to go in a different one. That was like 5 years ago, so here we are 5 years later. And he's talking on stage, and I'm talking on mic, but that's just the way life is. So I'll try to just be, objective about this, because I really don't care in the grand scheme of things what others people think. I just do want to document, though, what happened for this thing, because I think it's a good life lesson, about the way cover-ups work. A lot of people have this idea that there's some grand conspiracy or some, cabal of people in a smoke-filled room. And no, it's the do-gooders on the debate stage that cover these kind of events up. And I think it's a good time and way to document the way these things work. I mean, there's one thing that I learned from all this. It's, beware of the firefighter, because they're often the arsonist.
Engagement with complex questions. It's exactly the kind of discussion that this series is meant to encourage. Please join me in welcoming Matt Ridley.
I like Jay. I did not know about him until the pandemic. He wrote a piece about asymptomatic spread being, in other words, more people were infected than were showing up on the PCR test. So this thing has got a less of a lethal rate than most people are imagining. And that's when I learned of him. He was actually one of the first people to prove this thing may be a self-spreading vaccine, because it's highly contagious but not very lethal. Now, to get that to Jay is going to be, impossible when you got people like, Matt taking the stage. It's just the way it is.
How are you, Matt? Welcome. Thank you, Jay.
Ladies and gentlemen, let me say what an enormous honor it is to give this lecture today.
I've read both.
Defense of scientific
books.
freedom.
I've read Viral.
And I pay tribute to Jay Bhattacharya's courage in championing
it.
this issue and indeed in inviting me.
So it's on my blog.
In what follows, I have relied heavily on my co-author, Alina Chan, but if I get anything wrong in the next hour, it's my fault, not
completely
hers.
failed.
I'm going to explain why I changed my mind, why I went from dismissing the lab leak hypothesis to concluding that it was not just probably, but almost certainly correct. This was not driven by motivated reasoning. Quite the reverse. I am pro-science, pro-biotech, pro-genetic engineering. Indeed, that was one of the topics on which I spoke most often in the House of Lords. Although
Oh, I didn't realize he brought that up. I need to play that. Oh, wow. He must have seen me post that. Caught off guard here by that. Pimping was the thing of the word I used. This is interesting. Beginning of the outbreak, here he is. This is
The nation, less medication and less suffering. What's not to like? But commercialising that animal in the UK is currently all but impossible.
This is
My lords, looking further ahead, the same
February
scientists at the Roslin
1st
are now
of
looking
2020.
into how to control grey squirrels. Not by killing them, as we do now, but by using genome editing to spread infertility infectiously through the population.
He said infectiously. So he's conflating CRISPR, which is gene drive stuff, through sexual reproduction, which doesn't spread infectiously. But when he used the word infectiously, he's actually conflating that with a self-spreading vaccine. So that's, there's two competing thoughts in biology in early 2020, late 2019. And they're two different groups of people that are going at it, basically, and competing for big money, big government funds. And one of them is doing CRISPR gene drive research, which Matt Ridley, I would assume, is talking about. But he's so confused by the two different elements. He's talking about, actually, when he said the word infectiously, he's talking about a self-spreading vaccine. Matt Ridley is talking about a self-spreading vaccine in the House of Lords on February 1st of 2020. We're not even really talking about the COVID pandemic at this point, but that's basically what COVID was, a self-spreading animal vaccine. And he's talking about it. This is, how weird this world is that we've inhabited here. But when you're in the biotech space, which most lab leakers are, you kind of have overlap.
So that the population slowly declines while squirrels live happily into old age. This technique, called gene drive, is going to transform the practice of conservation all around the world, especially
the word infectiously is what,
control of invasive alien species.
So that just gives you an idea. He's in the House of Lords at the beginning of 2020 talking about, what this thing really was in the grand scheme of things. So I'm not getting into my theory here. I'm just going to focus on his.
I am a professional writer. I do have some relevant expertise in this matter. I have a PhD.
I was hoping, by the way, he would have taken this theory and run with it. I genuinely was. I never had any interaction with him. But I always thought, I admired him, and I was like, man, maybe he'll run with this. I'll just blog about it, basically, and he'll catch on and move on with it and run with it. It would be great if someone like him would have taken this, vaccine theory, which has become a fact in my opinion, and run with it. So, no, but he won't. Why? I'm not going to get into it why. I'm just going to try and stay away from that. I can't get into another man's mind as acting kind of irrationally from the irrational optimist himself.
In biology, I wrote a bestseller on genomics, and I was a member of the science.
Anyone that gets on stage and talks about themselves so grandiose,
but even if I had no expertise,
he doesn't have much evidence.
As a human being who experienced the pandemic, I and everybody else has a right to investigate this question. This is not something
beware of the first person singular pronoun too.
And I am frankly dismayed by how the WHO, most of the scientific establishment, including some of the NIH and the science journals, plus most of the mainstream media and some of the intelligence community, not only failed to investigate this issue properly, but were reluctant even to discuss and debate it. If a plane crashes, we do our best to find out why and share the lessons. This thing killed 100,000 times as many as a typical plane crash. As you will hear, by far the most important discoveries about what happened were made by amateurs working in their own time and on their own dime. So it's worth, I think, starting with the question, why does it still matter 6 years on? I think it matters for several reasons, because we do owe it to those who died to find out why. We need to understand that last pandemic. We need to explain why the virus was unusually infectious for a virus that had just intruded into the human species for the first time.
So I want to stop here, and that's really his only evidence he's ever proposed for a lab leak, that COVID was somehow pre-adapted for humans. No, it was pre-adapted for mammals, of which humans are one. There's 5,000 mammals on the planet, of which we're one of them. So COVID was pre-adapted for bats, which are not birds, they're mammals like you and me. So when the things, jumped species in the Wuhan BSL-4, it literally was pre-adapted for spreading in the upper respiratory tract of a mammalian bat, which is very similar to us humans. We have a similar ACE2 receptor. We have mammalian, bats have mammalian cells, so they also have furin. So the thing was pre-adapted for bats. That's the problem that he's having. He sees COVID circulating in humans, and he thinks it was designed for humans. And I'll get into that more later, because I think he brings it up a lot. But that is his main evidence. I've read his book, and he used the word pre-adapted several times, almost a dozen. And why? Because, he'll go off on that. Sends a lot of people, including himself. You would think he has the intellect to get over that. He doesn't. But he goes down this bioweapon warpath. This thing has been designed for humans because it's circulating in humans. No, it was designed for bats. It's a bat vaccine, a self-spreading bat vaccine circulating in humans. So apparently the higher up the intellect ladder you are, the harder you have a chance of getting that. So ironically, if I go into a right-wing website that's republished one of my articles, I'll go into the comments to see where people are. And, some right-wing dude's more likely to get it than, some high-mighty guy like this. But I don't want to get too far.
We need to prevent the next pandemic by learning lessons from the last one. And importantly, if we shrug our shoulders and say we never know.
There it is. He just, he took the words right out of my mouth. He thinks, he doesn't say it on stage, nor does he say it in the book, but he talks about, various PLA members of the science entity, not in Beijing, or not in Wuhan, they were in Beijing. Peng Zhu, I think is the name he keeps bringing up, who's basically a PLA scientist. So he's inferring that this thing was a bioweapon, because it's circulating humans, because he thinks it was designed for humans. So I'm going to try and limit my, rant on that.
We may never know how this happened. That sends a message to bioterrorists that they can do this and get away with it.
This thing has nothing to do with bioterrorism. COVID has infected us all, 3 or 4 times over. It has a 99.9% survival rate. It ain't a bioweapon. Okay, go touch grass if you think that to be true.
Attribution is deterrence. Not that I'm implying this was an incident of bioterror, but I think the next one possibly could be. In my talk, I'm going to cover the following topics: the outbreak, the market, the bat viruses, the connection with Laos, the lab, the fearing cleavage site, the diffuse proposal, and some loose ends. We're talking about a coronavirus. There are four kinds of coronavirus: alpha, beta, gamma, and delta. There are four coronaviruses that cause versions of the common cold and are endemic in human species, two alphas and two betas. There are two coronaviruses that have caused brief and lethal outbreaks in human beings in the current century, and there is one that has caused a pandemic. It is a close cousin of SARS. It is in the group known as SARS-like beta coronaviruses or sarbecoviruses. What is the natural habitat of sarbecoviruses? It is the guts of one genus of bats, the horseshoe bats, which live only in the old world, not in the new world, are highly gregarious and tend to live in natural caves.
So COVID doesn't infect old world bats, but it does infect new world lab bats. That was the first clue that this thing is something different. I'm not going to check into that here. I'm going to stick to his theory, not mine.
So the question we have to ask ourselves is, how did a bat virus get into people? And ignoring possibilities like frozen food, which are not very plausible, I think there are four plausible routes. The first is that it was a repeat of SARS, in which an intermediate mammal host caught the virus and that infected people. The second is that the intermediate host was actually a person who went into a cave and got infected. The third is that the person was a scientist. And the fourth is that the scientist took the virus back to a lab and did research on it. And the reason we need to take that last possibility seriously at the very start is because of the extraordinary coincidence of place and time. In all the many thousands of cities in China, and next to the 40,000 food markets in China, this outbreak happened in the very city where research on sarbecoviruses was occurring more than anywhere else in the world. That is quite a coincidence.
So he's not really answering the question, why Wuhan? There's really only, one person on the planet that answers the question, why Wuhan? And his name is Ralph Baric. In 2018, he wrote Diffuse. Ridley won't really say that. He'll say they. He'll go collective when it comes to accusations on Ralph. But he'll go singular when it comes to accusations on Shi. But I think it's theological. But anyways, I'm not going to get into that. This is Ralph Baric in 2018. I have no bat colony. No way for me to do colony. No way for me to do the experiment. He's trying to create a bat vaccine. My understanding is another colony exists in China. So here he is in March of 2018 talking about the Chinese bats at the Wuhan Institute of Virology. And here he is, in draft form. Here's Barrick's comment. We could provide this to you. He's talking to Linfa Wang. And then John Epstein replies, yes, we can do this with our Chinese colleagues at the WIV since they have access to the bats. So Ralph Baric answers the one question no one else can. Why Wuhan? That is it. That is it in a nutshell. And that was written by Ralph Baric in a document called DARPA Diffuse. And it didn't come out till 2021.
It's also a coincidence of time as well as place. In all the thousands of years of human history, this outbreak happened at the time when research was going on these kind of viruses for the first time. And lab leaks do happen, and have happened. Just to run through some examples of recent and relevant lab leaks, there was a global pandemic of flu in 1977 that was derived from an inadequately attenuated influenza virus used as a vaccine. There was an outbreak of anthrax in 1979 in the Soviet Union.
Christian Anderson did a paper on that recently and admitted to the 1977 lab leak, so pretty much everyone admits that now.
Which it took the Soviet authorities 12 years to admit was the result of a leak from a biowarfare lab. In Kunming in 2003, hantavirus infected researchers, and the person who investigated that case was Shi Zhengli of the Wuhan Institute of Virology. The same year, in both Taiwan and Singapore, researchers got infected with SARS while working on them, and again in Beijing the following year, at least twice, possibly four times, there was a leak of a virus. The important thing about those cases is that except for the Taiwan case, they didn't know how it happened. So these things can happen without anyone knowing that there's been an accident in a lab. In Surrey, in England, in 2007.
So, yeah, lab leaks occur pretty much every day, all the day, every day, all the time. It's not really a big event, in my opinion. So it was never the question of lab leak. That's not the debate. It was what leaked. What the fuck? Whipped around the world in a matter of weeks in early 2020 and shut down the world by March or April of 2020. So that is what made this thing so different. It was the contagion, the aerosolized agent that made it so interesting. It obviously leaked. The question was always, what leaked? What is that thing? So why was it so novel, so new, and so infectious? So that was always the question to answer, in my opinion. So it's not lab leak, it's what leaked.
Disease broke out on a farm just down the road from the world's reference lab.
And if we answer that question, we proved lab leak.
A coincidence, a contractor had
but he wants to get into the debate of lab leak.
And in 2019, just before the pandemic, brucellosis broke out and infected a large number of people in China
want to get up on stage and debate each other.
And finally, of course, SARS-CoV-2 has infected people in the lab since the pandemic began, probably many times, but we know of one particularly clear case in Taiwan. All in all, in the first 20 years of this century, there have been hundreds of individuals infected in scores of labs and in many different incidents. And just before the pandemic, the World Health Organization and organizations like the EcoHealth Alliance were warning that this lab leak risk was getting worse. The threat from lab-enhanced viruses is intensifying, and curiously, these warnings have been much quieter since the pandemic. That seems to me curious. Nonetheless, when the outbreak happened, the Chinese authorities immediately assumed they were dealing with a repeat of SARS, and that the Huanan seafood market in particular was where this was happening, and they went as far as saying that there was no human-to-human transmission, and they continued saying that up until the 20th of January, which turned out to be bad advice. In other words, the only way you could get this virus in January, they thought, was from another mammal. And at first, the evidence does look quite interesting, because the cases reported to the World Health Organization from December 2019 do cluster around the market, and that's more true of the ones that had no connection with the market than with the people who had actually been to the market. But that is largely a case of ascertainment bias, because the criterion for whether or not you could even be considered for having this disease in December.
Just stay away from the wet market debate. Okay, it's lineage A and B that they're debating, and WA1 is older. So, yeah, just avoid that thing altogether. The question isn't the wet market. The question is why Wuhan, and only Baric answers that.
2019 were that you had to either have been to the market or to live near the market.
For reference, the BSL-4 is down here of Daniel Anderson.
We now know
the
that there were at least
BSL-2
73
was
and possibly
a
as many
little
as
bit up
86
the road,
cases that were not reported
and the wet
to
market was
the World
across
Health
the
Organization
border.
from December 2019, and we don't know the origin of them. And indeed, according to a Chinese government document seen by the South China Morning Post, there were 9 cases in November. These have not been shared with the World Health Organization. They could be very important. Furthermore, the virus that people caught in the market was not the ancestral form of this virus. It was a derived form. It was the B strain of the virus. And that's quite an important piece of influence, because it's very hard to see how that could imply an origin in the market. There were mammals for sale in the market.
I don't think he's ever brought up the WA1 ancestral strain, ever. And nor will he ever. I don't even think he knows it exists. So essentially an American traveler, an American, went back to Wuhan, I guess to renew a visa. He was there for like 6 weeks, and he lives in Seattle. He may work in like a tech industry. And from November to January of 2020, he was there in Wuhan, but he never visited the Wuhan market, and he came back to Seattle, checked into hospital sick, and he was the first confirmed case of COVID in America. So he's America's patient 1. And the CDC took a sample of his virus, of his isolate, and then in that process, we learned a lot, which lab animals made this thing. So I don't get into that theory here, but there is the ancestral strain was collected by an American traveler. The oldest known strain, predating lineage A and B in the wet market, was collected by an American traveler. It is essentially the proof for a Wuhan lab leak, but at least U.S. lab origin.
And in other markets in Wuhan, not very many by the standards of markets in southern China, where the sale of live wild mammals is much more common, but there were some, and they
This is all
animals
water
that
under the
could be
bridge.
susceptible to this kind of virus.
He has mink neovicin on there. I'm not sure. That's never been documented in the wet market, because they're not even in Wuhan. They're not even in China. So that was a big clue, though, that the mink were infectious to COVID. Infectious means onward transmission. Infection by transmission is the way I put it. So you can infect a raccoon dog, it doesn't infect a raccoon dog. You can infect a hog badger, whatever the hell that is, and it doesn't infect another hog badger. You want onward transmission, ideally without symptoms. So it shows a reservoir host situation when it does that. But I don't know why the heck he got that. There is no mink in the Wuhan market. Even if there is, it doesn't really matter.
But it did not include bats, it did not include pangolins, and all of the raccoon dogs for sale, for example, in this market were sourced locally. They had not come from Yunnan, which is where this virus had come from, or southern China generally. It is true that virus was present in the market, not just in people, but also on countertops.
I'm going to kind of fast forward through this. I'm really bored of the lab leak debate when it comes to the wet
in the
market.
southwest corner of the market, which is where those mammals were on sale for sampling. So again, this is almost certainly a case. Mammal
This
DNA
is just dumb.
and
They're
virus
looking at
RNA,
stalls
you
in
find
the
a
wet
negative
market. That
correlation.
is just
In other words,
completely irrational.
that has both raccoon dog DNA maps to the virus. That is not strong evidence for the CDC to announce in May of 2020, quite early in the pandemic,
George
that he
Gao.
thought the virus was not the source, it was
Look, I
more
don't
like
like him.
a victim. And it was this.
I think he knows who I am. And I think he knows of the blog and the theory. He's too close to this not to know. But I'm not going to speculate on that.
Press announcement,
What does he know? I think he knows about as much as we do. Not much.
rather obscure, but I did stumble upon it, that led me to begin to change my mind. I was gobsmacked by this. I thought it was case closed at that point, and I began to go back and look at the evidence more closely. I can't stress enough how different this is from SARS.
George Gao, by the way, has pointed out the North American white-tailed deer in one of his published papers. So he's thrown some subtle shade at Fauci and the NIH for maybe cooking this thing up.
In the case of SARS,
But if you don't know my theory, that's basically it. This thing was cooked up in a NIH lab, Rocky Mountain Lab, to be specific, along with Ralph Baric's help at North Carolina, and it was made as a transmissible, self-spreading bat vaccine, an aerosolized bat vaccine. So you just take the needle out, you infect one bat with it, and it spreads autonomously to the next bat. That's what COVID is. That's what it was. So, and that happened, that the jumping of species happened in the BSL-4. So if a virus is engineered, wherever it spilled over in the human population, does not tell us where it was engineered. I'm going to say that again because Matt will never get that, never will ever say that. So if a virus is engineered, wherever it spilled over into the human population, does not tell us where it was engineered. So we're debating more of a lab origin than we are a lab leak per se. So you also have to separate the two. Yes, there's a lab leak in Wuhan, but there's a lab origin in the U.S. tied to it. So once you dive into the weeds of these MTAs, material transfer agreements, and emails that they all do, these virologists live in a very unique world where they share samples and share vaccines and share trials with one another. It's very niche.
Infected civet cats, and in particular,
It's their
people who
currency.
handle such mammals, were very clear from the start. In this case, not only were no animal handlers infected of a relevant kind, not one.
Yes, we agree. So we're going to go through this.
Of SARS-CoV-2. What do we know about?
There's never been a reservoir host found by the Zunati, by the natural origin scientist. We have them. They're just not in
This
Wuhan.
virus? Well, when Shi Zhengli first published the sequence of the virus in Nature, she revealed something very important. There were 3 bat viruses that were closely related, one of them particularly so, RATG13. And this is what she said about it. We then found that a short region of the RNA-dependent RNA polymerase gene from a bat coronavirus, which was previously detected in Rhinolophus affinis from Yunnan province, showed high sequence identity to the new virus. We carried out full-length sequencing on this and found it was 96.2% the same. There's 2 features of that passage of text that I need to draw your attention to. The first is that they clearly imply that they sequenced the bat virus after noticing the similarity between one of its genes and one of the human virus's genes. That turned out to be false. They had sequenced the bat virus in 2018, the year before the pandemic. In which case, why did they need to notice just one of the genes being similar? Why didn't they blast the sequence similarity of the whole genome? And the other...
He's going off into a conspiracy that's kind of been old and debunked. I don't know if I really want to do it here because it's so bizarre and into the weeds, but... I don't know how to say this really quickly without being too belittling of it, but where is it? This came out later, so this later is 2022, so that's 4 years ago. I don't think Ridley even knows about this, or if he does, he just conveniently forgets it. But Shi Zhengli of the Wuhan Institute of Virology uploaded a partial sample of RATG13 to the NCBI, that's the NIH database. So Shi Zhengli has shared RATG13, not all of it, but a little bit of it, with the NIH before the pandemic. It's under embargo, so it wasn't released for 4 years. So she did it in 2018, and it's released in 2022. So this is concerning. Let's just read what Francisco said here, who both Matt Ridley and I like. NCBI had a 98% and a 97% match with SARS-2, and we didn't know about this for 2.5 years. So if there's a cover-up here, it's actually on the NIH side. It was under embargo. It's not really that weird. When you get into the whole weird thing of publication, they embargo these sequences until the paper's published. But for whatever weird reason, the embargo was lifted, I guess automatically, and then it published in 2022. So that was the mystery solved there. Shi Zhengli, yes, she sequenced RATG30 in 2018 to upload it to an NIH database so that theoretically someone like Ralph Baric could download it. So in other words, Shi Zhengli shares samples with Ralph Baric. She shared SACO14 with them. She shared another one called, what was it called? I'm already forgetting the name of it. MERS 422, something like that, I think. This one was called 4991 before it was renamed. So there's nothing really nefarious there. But this is what happened, is Ridley started off in the Leibniz debate so early, he got bogged down in these deep rabbit holes before diffuse leaked. And I came a little later, around the time diffuse leaked, and I I guess I'm going a different direction than y'all are.
The strange thing is that notice the sentence about the previous...
So in other words, he doesn't have any hardcore evidence. It's just speculation. It's just conspiracy on top of conspiracy. He doesn't have any... I can go to a lab leaker like Ridley and I say, give me one piece of evidence for Wuhan lab leak. And he'd probably go fear and cleavage site, and I'll tell him, well, Shi Zhengli's virus cells deleted. And then he'll go database. Well, we'll talk about that. And I'll show you publication of RATG13 kind of eliminates the database need because it's a 100% match. So this is what happens when you come too early to the party. You're not the life of the party. You want to show up right at the right time, right about midway through, and everyone thinks you're great because you just showed up at the right time. It's all about timing.
Does not have a reference.
Life.
Does not have a footnote. Does not have
Don't be the first guy at the party.
I spent several
don't be the last
days trying to track down RATG13 on the internet. When had they found it? Where had they found it? Under what circumstances?
It did not exist. Getting all worked up over this.
It took us more than 6 weeks to resolve this.
Very emotional over Shi Zhengli.
That did so was the result of Mira Helka's work in India and Rosanna Segretto's in Norway. And what they noticed was that there was a virus out there that had been described in 2016 that had a 100% similarity to RATG13, but it had a different name. It was called 4991. And it later transpired that they had changed the name from 4991 to RATG13 at the start of 2020.
No, they did it in... see, this is so deep. I didn't realize this is my first time to watch it, all the way through. So they renamed it RATG13 in 2018. Shi Zhengli has renamed RATG13, renamed 4991 to RATG13 in 2018. Here it is. It's called RATG13 when it was uploaded in, July of 2018. She has uploaded it as RATG13. So, this is just, the guy's just going to keep lying and lying and lying. That's the only reason I'm doing this. It's annoying. The guy obviously doesn't even know about any of this. If he does, he just ignores it. It's his own guy. He's already referenced Babar and Francisco that found it. So, it's an inconvenient piece of evidence. Here it is, RATG13, uploaded in 2018. Not all of it. It's just a little short sequence, but it's something. And I don't know what happened, to be honest with you. Only her and Baric know, but I'm just assuming Baric was like, hey, this will be of interest to me. Why don't you just give the whole thing to me instead of piecemealing it up to the NIH database? So that's the one piece that is missing in the whole equation here. But you see, he's gone off on this tangent now that, she renamed it after Lab Leak from 4991 to RATG13. She renamed it before Lab Leak to RATG13. RATG13 is the Rhinolophalus bat, Tengguan province, and 13 was the collection date in 2013. And it was funded by the NIH. The NIH funded the collection of RATG13 in 2013, and the NIH was sitting on RATG13, parts of it at least, in 2018. So if there's a conspiracy here, it's what's going on with the NIH funding the collection of this thing, having it sitting in a database and not releasing it for several years into the pandemic. So it's just, it's China, Jim, so shut up.
Not before, but hadn't communicated this fact. Now, whether intended...
The self-righteousness, too, of this guy is kind of old for me, too. I did like him, by the way, and, I'm not going to say I dislike him. I don't dislike anyone in the grand scheme of things. We're all a bunch of fucks trying to get by in life. But this is the way cover-ups work. I just just document. Don't get too worked up on it myself. But I don't like the guy. I'll be honest. I just don't. I did, but I don't anymore because it's a bit annoying. We get this close to the finish line, and we got this thing more or less proven. I mean, I got Ralph Baric's name in Nature with SAGO and the World Health Organization, and he wants to get up there on the debate stage and talk about Shi Zhengli. It's a bit bizarre to me. Because it's not about lab leak for him.
Disguise the story of the closest related bat virus and to make it rather obscure at a time when it was quite possible that the pandemic would blow over as only a minor event.
Again, she uploaded RATG13 in 2018 to an NIH server.
Well, the first thing is, it doesn't come from Wuhan. It doesn't come from anywhere near Wuhan. It comes from southwest China in Yunnan. It comes about as far away from Wuhan as the Florida Keys is from where I'm standing today. That's a long way for a virus to travel. And we know of only one animal.
They travel farther than that, For example, SHC014 was collected around the same area. And it went back to the Wuhan Institute of Virology, where Shi Zhengli emailed it halfway around the world to Ralph Baric. And Baric downloaded the genome sequence of SHC014 and turned it into SHC014 MA15, mouse-adapted 15 passages. So Shi Zhengli goes out there to the damn bat cave, because she's really small, unlike Fat Ralph, and she can crawl in there under her little skinny, hiney, and get deep into those caves and get that bat sample back into the Wuhan Institute of Virology, where she just has to, put it in her Illumina machine, which is not even new, it's used. And then she can just upload the sequence to the database or email it to Ralph Baric, who can download the sequence and produce a live virus in Chapel Hill, North Carolina, from a bat sample that was collected in the middle of nowhere, China. So that's the story here. It's not, from here to the Keys, who cares? And when you email these sequences around the world. So viruses and samples are shipped around the world. Sometimes, a lot of times physically, frozen ice, 48 hours. Baric's biotech, by the way, is so advanced it fits in a 1 millimeter tube and sits on dry ice for 48 hours. And it can be readily available anywhere at any time. And Shi Zhengli's biotech is basically, her just being able to upload the database or upload the sequence of the database for someone like Baric and his grand biotech to download it. So this is not the story here from here to the Keys. The story is the transfer of these across continents and whatnot. So it's not really nefarious, at least pre-pandemic. It's what they all do. It's what you would do if you're in the industry. It's very international, by the way. So your nationality is not really seen as a hindrance, in this case.
That has made that journey from a bat cave to Wuhan. It's Homo scientificus. So what is the story? How had they found this bat virus and under what circumstances? Well, we did actually have a report in the scientific literature from 2014 that was around...
He's going to go off on an RNTG13 conspiracy, the Moijang minor conspiracy. That it kind of comes to a dead end when you get to the Laos sample.
And the events that led up to the discovery of that virus. This article is actually about a different virus, a rodent virus, but nonetheless it's
He's
about
basically
the same
an old chap
but it
stuck
wasn't
in
until
2020
May of 2020 when a young Indian named the Seeker found a medical thesis from Kunming Hospital that gave us the details of this story. 6 men had gone into a disused...
He's the only one still talking about this. No one else does. It's kind of old
copper mine
news.
on a mountainside in Mojiang County in southern Yunnan to shovel bat guano out, probably with a view to reopening the mine. They had got sick, badly sick, with pneumonia. 3 of them died, 3 of them eventually recovered. And this was a case...
It is an interesting story, but it's a dead
China
end.
to get very interesting. Samples from the men's lungs were sent to Wuhan and were found to be positive with antibodies for SARS. And that led to the conclusion that these men were probably the first and only occasion where human beings had caught a sarbecovirus directly from a bat, which would be a very interesting story in its own right. And that led the Wuhan Institute of Virology to mount a number of expeditions to the site, about 7 over the next 2 years, in order to sample the bats in the mine shaft. And you can see...
This event was referenced in the NIH grant CREED, which more or less funded Diffuse, but they talk about the dead miners. In the CREED grant by the NIH to Ralph Baric, Peter Daszak, Shi Zhengli, and Linfa Wang, it's referenced in the NIH grant to them. This event interested the NIH for the reason he's bringing up. He makes it sound as if it's some sort of, Chinese conspiracy, but it's more like an NIH interest. This is what the NIAID in particular does. Infectious diseases.
See them here dressed in full PPE.
Which all come from bats, which is why they were vaccinating the bats.
And handling live bats in a keep net. We'd like to know more about this site, but journalists and others who've tried to get there since the pandemic began have been turned away often very firmly. And there has been nothing published, literally nothing, about research from this site ever since. We've had no reports of whether or not there are still infected bats there or something, which seems a little strange, to put it mildly. So these are some of the things we need to know about 4991 or RATG13. Why did they change its name?
They changed the name in 2018, not 2020.
Why did they not cite their own 2016 paper in which they described the RDRP gene from it?
I don't know if I can answer that, but that paper is funded by the NIH. I'll leave it at that.
Why did they mislead us about the date on which they'd sequenced its genome? Why did they omit a connection with...
They really didn't, because Francisco figured all that out. He went in there, and look. He's just a genius in that stuff. You can tell when they sequenced it. It was all 2017, 2018. I don't know what his deal is.
A human disease.
I gotta,
and why did they claim wrongly, as it turned out, that there was no more sample after we finished the genome sequencing?
I believe that. Basically, she couldn't isolate it. Shi Zhengli is on record. I could not isolate it. Isolate a virus is a completely different thing than sequencing. So you can sequence any virus, more or less. You just stick it into the super duper Illumina machine, and it spits out a code, a sequence for you. So an RNA sequence is pretty easy to find and get. But isolating a virus, creating an infectious virus, and bringing it to life out of bat guano is borderline impossible. And the Wuhan Institute of Virology is pretty much the only people that can do it. And they've only done it with three samples. So RATG13 sounds a lot like SHC14. That was another sample Shi Zhengli couldn't isolate. So it's pretty worthless to her. She can't isolate. She can't grow it in her vira cells. So she emailed it to Baric and said, hey, have fun with this one, because I can't do anything with it. Here's the sequence. You do something. And Baric could download the sequence, the SHC14 sequence, create a live virus. Something similar happened with RATG13. In RATG13, before I forget about it, is 25% different than SARS-1, which is precisely what Baric was looking for in his bookend in DARPA Diffuse, 25% deviation from SARS-1. Why? I'm not going to get into that here. You just ask him and he won't answer, but he calls it his bookend.
In the summer of 2020,
here they are, Babar and Francisco.
Two sleuths, one called Babar, one called Francisco.
They're the ones that figured this out.
Spotted
in 2022
database that there were 8 other viruses that had apparently come from the same site.
He's going off on these.
And were closely related
we'll come
to
up with that later.
We eventually,
lineage 4 stuff.
This was eventually confirmed to us by Shi Zhengli.
These are not close to SARS-2. None of them are closer than RATG13.
And her colleagues. Yes, we found 9 viruses altogether in our expeditions to that mine, not 8. Which means that the Wuhan Institute of Virology was working with the 9
he's calling it lineage 4.
closest relatives to SARS-CoV-2.
Well, I'll give that here, I guess. Where is that? Lineage 4. I gotta have it on here somewhere. I thought I already had that one. Here it is. So he's already called it lineage 4, essentially. These are lineage 4 viruses, and I'm not going to do it here. I'll save it for later. He brings it up again, but this is how conspiracy theorists.
The way he does.
He's got so deep into this secret conspiracy and the thesis written by a friggin' postdoc, as if a postdoc created COVID. The most infectious disease on the planet that we're still debating 6 years later is quite preposterous to even assume, but teach their own.
Thesis, and which shows their relatedness and includes 4991.
Ping you. It's not even. I don't think other one
was published at the end of 2019, which is how we know they hadn't yet changed.
Maybe 22. I don't know.
But when Ping Yu's thesis was published as a paper in Infectious Genetics and Evolution, lineage 4 was omitted altogether. That may have been because they were saving it up for a later publication or something, we don't know. But it does mean that we didn't know that they had lineage 4 in their possession at all.
He goes collective with his language. If he wants to use it for his own intellect, I'll use it against him. So we don't know anything. There is no collective knowledge in this thing. It's individuals presenting evidence, and he goes into that collective. We know. It's usually when someone's trying to bluff, they don't have anything. And it's a dead end because RATG13 is closer than all of these.
Or what they would.
And my contention is, before I forget, if you believe Shi Zhengli of the Wuhan Institute of Virology published RATG13 and then inserted the fear and cleavage site into SARS-2, I think you're pretty dumb. So here's RATG13 at the top, gap. Other than that, it's 100% amino acid match for 500 amino acids right here at the S1S2 border. And here's SARS-2 at the bottom. It's pretty easy to see what's going on here. So when Shi Zhengli published RATG13, she exposed the fear and cleavage site. We'll probably come back to this, but it's very important and try to just drive that home. That Shi Zhengli has given up a piece of evidence proving SARS-2 is engineered. So whether you think she's innocent or not, come to your conclusion, but I'll come to mine. But she has given up a piece of evidence showing that something's going on with SARS-2.
Then in 2021, there was a complication in this story.
The Laos
because
connection.
The Pasteur Institute found a virus in Laos that was more closely related to SARS-CoV-2 than RATG13.
So
96.8% rather than 96.2%.
He knows that was collected by the DoD.
Well, that seems to change the story. But it doesn't necessarily, because
The U.S.
neither of these viruses is the
Department of
ancestor
Defense
SARS-CoV-2.
went
They're
out
not
there
close
and
enough to be
collected
the
Laos.
progenitor, but they are representatives of the group from which.
A closer, it's not even easy to comprehend, but in 2017, the U.S. military collected a sample from Laos, just south of China up here, that is closer to SARS-2 than RATG13. And it was collected by the U.S. military, not EcoHealth, not Shi Zhengli, not the Wuhan Institute of Virology, not any China scientists. U.S. military funding collected the closest known progenitor in 2017, and we didn't learn about it until 2021 because they couldn't publish the results. They had to go back out there, the local lab from Laos is sometimes pronounced, to recollect the same samples from the same bats without the DoD money. So we don't know what happened to the original samples, but it's pretty obvious if you don't, whenever the Uncle Sam's tax dollars fund something, they get the sequence. You'll find that a lot. Every sample that Uncle Sam sells or funds comes back to usually DC, goes into Ralph Baric's inbox. And, it's used to create countermeasures.
This virus is derived. And besides, the fact that they come from a different country doesn't necessarily change anything because...
So, let me back that up real quick because that's an important slide he's got there. So the U.S. government collected or funded the collection of banal in 2017. This one is, where is that? I should know where that is. That's Vietnam. That's Cambodia. So that's Cambodia sample. I think it was collected in like 2011 or something, if I remember right. With NIH funds, another one, and that's in Thailand. That would be, collected for the pandemic, all with the NIH money. All these other ones collected with NIH money, you'll find, a bunch of tax dollars, U.S. tax dollars attached to these bat samples, because it's, there's a bunch of U.S. military bases along the Pacific Rim right here, in particular Japan and Korea. So, and down here in the Southeast Asia, obviously encircling, China. So, there's a special interest between virologists and the military that always overlaps with these viruses.
The fact that they come from a different country doesn't necessarily change anything because the Wuhan Institute of Virology was receiving viruses from Laos via the EcoHealth Alliance. Yeah,
he's done a bait and switch here. I don't know if I can get into it here in the video, but the samples they collected from Laos were at the border with China. And the Laos Banal sample was at least, a couple hundred miles away. So you can see where it is. He's found some samples collected up here by the Chinese border, and it was down here where the actual closest known progenitor SARS-2 came from. That's like, chapter 19 or 20 of my book. It's on my blog too if you want to go there. But this thing is one of the weirdest little twist in this whole story. But he knows it too. The U.S. DoD, DARPA's preempt program, is more information on this. Looks increasingly like, how did I get there? Blah, 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 blah. I mean, he knows about this, but he's playing dumb there up on stage because it doesn't fit his narrative. So I'll get rid of that, and we'll keep going with this.
As shown by both emails and research reports, and indeed genetic databases. This one shows Peter Daszak depositing a coronavirus at the Wuhan Institute of Virology from Laos. So viruses were crossing international borders here without report, which is of itself quite interesting. When the outbreak happened, Shi Zhengli gave an interview to Scientific American, and she said, "I have never expected this kind of thing to happen in Wuhan." What did she mean by that? She meant that they had been sampling bats in the vicinity of Wuhan in Hubei province extensively over preceding years and had never found these kind of sarbecoviruses there. So they'd also been sampling human beings in Wuhan and had not found SARS-like antibodies in them. So no wonder she worried, could they have come from our lab? She eventually concluded that they had not, but we'll have to see whether that's plausible. What was going on in Shi Zhengli's lab at the Wuhan Institute of Virology? Well, here she is with Peter Daszak of the EcoHealth Alliance in her laboratory. Peter Daszak was funneling millions of dollars from the US taxpayer to the Wuhan Institute of Virology to support her research. And this is one of the NIH grants that covered that. Understanding the risk of bat coronavirus emergence.
I don't know if funneling is the right word. He's just winning grants that are money that the NIH is giving away readily. Why? Less for her and less for him. It's more for Ralph Baric. Ian Limpkin's another name you'll see a lot. These big names in NIH research want these samples from abroad. And like Ralph Baric wants to travel out there to Timbuktu, China, when someone like Peter Daszak will do it. So they're basically just contractors for Ralph Baric and Tony Fauci doing the dirty work.
The work was intended to, Shi Zhengli had begun, obviously, by tracking down the origin of SARS. But the next step was to...
So this grant was renewed in 2019, and Ralph Baric, this is the 2R01, so he doesn't have the NIH grant number, right? The 2R01 was from 2019 to 2024, or whatever that is. So, yeah, he's not in the weeds. He doesn't know the details. But that 2R01 is what funded DARPA Diffuse. TA1 and DARPA Diffuse is funded by the 2R01 grant, which ran from 2019 to 2024. And Ralph Baric did that little switcheroo. I could bring that up. And,
identify other
I'm going to stick to this.
viruses that might cause pandemics, and then also to study them closely in the lab. This project did not receive universal support from virologists. Indeed, Andrew Rambo, on the 21st of November 2019, just as we now know the pandemic was starting, wrote on Twitter that the more we look, the more new viruses we find. The problem is we have no way of knowing which may be important or which may emerge. There is basically nothing we can do with that information to prevent or mitigate epidemics. So he's saying the benefit of this research may not be very great. And Peter Daszak replied to him, that's not true. We've made great progress with bat SARS-related coronaviruses, identifying more than 50 novel strains, sequencing spike protein genes, identifying the ones that bind to human cells, using recombinant viruses and humanized mice to see SARS-like signs. So live infectious experiments.
Self-righteous kind of voice right there. He does a lot of cropping, but underneath this is like a whole thread about Peter Daszak ranting about Ralph Baric, not Shi Zhengli. That's what they're doing all this for Ralph Baric.
Small, according to Rambo, the risk is suddenly sounding like it might be quite high. Is the risk-benefit ratio really right here? A few weeks later, still not knowing that a pandemic was brewing,
here's Daszak talking about
gave
Ralph
an interview to Vincent Racaniello in which he explained more detail what they were doing in the lab. Coronaviruses are pretty good. You can manipulate them in the lab pretty easily. The spike protein
to his credit, I think he brings up Ralph Baric.
You can build a protein.
It's
You
kind
work
of
with Ralph Baric
massive.
at the University of North Carolina to do this. Insert into the backbone of another virus and do some work in a lab. So if you're detecting viruses in bats, but you can't grow those viruses because they're not in a good enough state to get them into the lab and grow them, then you can at least extract their sequences and from that synthesize their spike gene, stick the spike gene like a cassette into another virus that you can grow, and then you've got a chimeric virus that you can test on cells in the lab. Now, Ralph Baric had invented these techniques and had collaborated with the Wuhan Institute of Virology to do this work. It would be nice to know what he knows. The University of
He is the creator of the SARS-CoV-2 genome. I have asked him for an alibi. He forwarded it to UNC Communications. They replied back to me, and they said, our statement is his testimony. And inside that testimony, I basically can convict the guy. He doesn't have an alibi. He is the creator of the SARS-2 genome. It was his pietà, his masterpiece, his lifetime work that leaked from that Wuhan lab. I almost feel bad for him in a way. I'm not going to get into why. But, yeah, I mean, imagine your life work just kind of, being shipped off to Wuhan by someone else and getting out and someone else is watching, not yours. And, yeah, there goes your whole life. But, he'll live. He'll be fine. But yeah, that was his masterpiece that leaked from that Wuhan lab. That's why it took so long for us to figure out where this thing came from. He was able to create something from nothing. I'm not going to get into that here.
North Carolina has
I actually kind of admire the guy. I mean, he's just, interesting.
steadfastly refused to share his emails and is fighting in the courts over that. And he himself...
See, Ridley apparently only thinks you're guilty once you start, not giving up stuff, which is what Shi Zhengli, claimed she wasn't doing. And now he's only suspicious of Ralph because he's been fighting in the courts with the U.S. Right to Know. But, yeah, that was inevitable.
has not given any evidence to Congress in open session. But he has gone on the record to say of the lab leak as a theory about the origin of COVID, you can't rule that out.
You can't rule out a Wuhan Institute of Virology lab origin is basically what Ralph has been saying. He played the lab leakers like a fiddle. He signed onto the science letter saying, hey, we can't rule out the BSL-2. And the whole time he was trying to divert attention away from the BSL-4 of Daniel Anderson. So Baric was in DARPA Diffuse sending samples to the BSL-4 of Daniel Anderson, not Shi Zhengli's BSL-2.
We do know what some of these experiments achieved. This is a tardy report from the EcoHealth Alliance that was eventually filed, which shows one experiment in this slide where they took the spike gene from a virus called SHC14 and put it into the backbone of a virus called WIV1, and the result was a 3-fold increase in the death rate of mice and a 10,000-fold increase in the viral load in brains and lungs of humans.
Baric has a great line. Are you trying to make viruses more dangerous? He goes, if you're a mouse, yes. I love his dry humor, and that's essentially what's happened here. And Shi Zhengli's caught on to his methods, and those methods are safe. Because COVID doesn't affect humanized mice. Never has, never will. I mean, it's to claim COVID infects humanized mice, it's also claim it infects raccoon dogs in a wet market. It's 100,000 infectious particles shoved with the nose of the poor mouse, and there's no contagion. There's no spread. So it's not really an infection model, essentially. That's what you do in a lab infection, and there's nothing there. So it's a bit preposterous to even claim that. But yes, Ralph Baric was able to push the genomic edge in humanized mice. This guy that wrote the Red Queen saw the word humanize in front of a mouse. He thought it was a mouse. And Baric's line is brilliant once you get it. If you're a mouse, yeah, I'm trying to make the thing dangerous. But we're not mice. We're humans. So there's nothing humanized about a mouse. This was, I gotta, finish my rant here, I guess. This is, where was it? I think it's up here, essentially, where I talk about it. Maybe not. Maybe it wasn't in this one. I wish it was. Well, we'll play this while I'm here. So I think Matt really scared of Ralph Baric while I'm here on this one. Maybe I'll find out what I was looking for. But this is him segueing during a talk recently to bring up Ralph Baric. He seems nervous. I've never seen a guy get a little fidgety like this before. He's just not comfortable talking about Ralph Baric on camera. He's intimidated by him for whatever weird reason. I don't know.
So that's one reason. We actually have a very interesting marginal footnote in one of the drafts of the defuse proposal, the one where they were talking about putting a furin cleavage site into
He's just really uncomfortable, isn't he?
for the first time.
I've never seen a grown man this nervous.
A grumble from Ralph Baric at the University of North Carolina, in which he says, hang on a minute, shouldn't these experiments be done at biosafety level 3?
He's really talking about his own experiments. I mean, I'm not going to play this whole thing and bore us with that. But Ridley is just, he's scared of Ralph.
That is gain of function.
Which to me is
There's
funny.
Absolutely no dodging that. Of course it is gain of function.
And Barrick's, to finish off on the humanized mice thing, to Barrick's point, as you adapt the virus to a mouse, it moves away from a human. It's less likely to infect us and our human cells. So it's a safe model for pushing the genomic edge and genomic research. So it doesn't matter if it was a millionfold increase. It killed the damn mouse, but it's not going to kill a human because humans aren't mice.
But this experiment is on a virus that is very closely related to
and it's
SARS,
not a contagious
not
model.
to
It's not a transmission model for SARS-2.
So it's not the experiment that could have led to this pandemic. However, what other viruses were they doing these experiments on?
These mice are so safe, Baric would send them to Wuhan. I mean, because what's going to happen? Nothing. I mean, the proof of the pudding is that there's been like 10 or 12 mice bites or incidents in Chapel Hill, North Carolina, and no one got sick. Baric doesn't even send them home with a mask. He just says, take the rest of the day off. You're fine. Nothing's going to happen to you. The damn mouse bit you. Who cares? It ain't going to affect you. I know what I'm doing.
And the answer is we don't know. We know which viruses they were working on up until 2016, but we do not know which ones they were working on in later 2016, 17, 18, and 19. So it's possible that they were working on ones that were more closely related to SARS-CoV-2 by then, that were in lineage 4. We probably would know if we could see their viral pathogen database, which they maintained and kept open to outsiders up until suddenly they closed it at 2 a.m. on the 12th of September 2019, a couple of months before the pandemic started.
I think it's online until February of 2020. Let me see if I can find that. I got all these lined up. Yeah, so there it is. It is online until February 2020. You had to be like, you had to have a special password, but it's still kind of on there until February 2020. It was online in, January 2020. So it did go offline later. But in the grand scheme of things, my argument is this is the database. Shi Zhengli has published the database right here for us to see. Because it's biologically impossible to publish something closer than RATG13, which is a question that I've asked Matt himself, Matt Ridley, hey, why did she publish it? And I've asked him that, probably 4 times in the past 4 years. I don't know what more I can do, but he won't answer it because his answer won't be good because he can't answer it. Because if you think Shi Zhengli published RATG13 instead of that furin cleavage site, I think you're a simpleton. I genuinely do. But anyways.
And they've never shared with it since. The reason they give for not allowing us access to that database, which would exonerate them in a flash if it has no relevant viruses in it, the reason they give is because they don't want people hacking it. But if you share data,
this
you don't have to worry
exonerates
about
them.
people hacking it. So I don't
this is the
understand
database.
That, excuse.
I have a better alignment
however,
somewhere
from
else.
a grant renewal document to the NIH from the EcoHealth Alliance that there was a plan to broaden the range of viruses they looked at, to move away from ones that were very close cousins of SARS to ones that are 10 to 25% different. The reason
That's Ralph Baric's terminology right there, by the way, not Shi's. Because that's what Ralph's always talked about.
Because the ultimate aim of this project is going to be to produce a vaccine that will work against any kind of SARS-CoV virus. Now, it's interesting because SARS-CoV-2 is 24% different in the spike gene from SARS. So this is exactly the kind of virus they were moving to take seriously from this point on.
Yeah, it's 24.7 according to Ralph Baric, the spike, but the whole genome is about 20%, and Baric basically patented SARS-2 in 2018.
When this sequence first appeared, it alarmed a number of Western virologists. Christian Anderson wrote to Anthony Fauci at the
on January 31st of 2020, that is 1 week after Shi Zhengli has uploaded RATG13. Notice how Matt Ridley won't bring up the date of when she uploaded it, which is January 24th of 2020. So here he is, almost incriminating himself and doing my work for him. On January 31st, we all find the genome inconsistent with expectations from evolutionary theory. It's a great line, because this is what Christian Anderson is looking at on January 31st of 2020. Shi Zhengli of the Wuhan Institute of Virology has uploaded RATG13 to a database called GIS-8. Because she had to go around the embargo of NCBI that was embargoed there from 2018 to 2022, and she had to go around the embargo and upload it to GIS-8, the competitor of NCBI. This is a great story, and she's getting the word out, like, hey, this thing didn't come from my lab, and it is engineered, basically. I don't know if she's doing it willingly, she's just basically covering her own ass. But when she uploads RATG13 on January 24th of 2020, a date that Ridley will not mention because it doesn't work with his conspiracy theory, Christian Anderson downloads it and does what's called the alignment, and he, on his computer, and California is looking at what we're looking at. You know, to be a rocket scientist to understand, PRRAR is the fear and cleavage site at the S1S2 border. Someone, like the hand of God, or Ralph Baric, has gone in there and split the genome apart, RATG13, and inserted a fear and cleavage site right in the middle of it. So, you know, it doesn't take a rocket scientist to do that. But you had to be, you know, like Christian Andersen, a world-class evolutionary virologist, to have all the computer, you know-how and have a GIS8 password. That was a real clue or a question. Was it when did Shi Zhengli upload RATG13? Was it before the February 1st teleconference? Yes. She started the February 1st teleconference by uploading RATG13. So there's no conspiracy here. Shi Zhengli has kickstarted
on the 31st of January 2020,
the Western cover, the
I
NIH
mentioned that after discussions earlier today, Eddie Holmes.
I like Christian Andersen, by the way. I mean, he's a truth seeker in the grand scheme of things. He just ran into a juggernaut of Ralph Baric.
Bob Gary, Mike Fazan, and myself all find the genome inconsistent with expectations from evolutionary theory.
He will not show the alignment and why he's saying that, because it doesn't fit Matt Ridley's conspiracy. This is what he sees as inconsistent from expectations from evolutionary theory, and he has a piece of evidence from Shi Zhengli, who has offered it up.
What did he mean by that? He meant that in the middle of the spike gene of SARS-CoV-2, there is something that sticks out like a...
To his credit, he's showing it now, but he doesn't show the full thing. You see, he's got this cropping thing. He's just kind of added it in there with all these others, essentially. But there is nothing even remotely close to SARS-2 until RATG13 is uploaded. So it's a bit, he's just such a sneaky little bugger. What SARS-2's closest predecessor or precursor was ZC45 and ZXC21. It was down here. So you had to be, really good at doing this kind of stuff to see it was engineered from that one. To even see there was a fear and cleavage site in it, because there's really not much of a match, especially on this S1 side. But none of these others were really in the database or in the system until Shi Zhengli uploaded RATG13, and that was the match. That was the close match. And he just kind of crops it off on the edge right here for reasons that he's just, it kind of muddies the water, essentially. So I won't go too far into the weeds on that.
A feature called a fear and cleavage site that massively increases the ability of this virus to be infectious in human beings, because it enables it to use a human enzyme called furin to cleave the spike and facilitate entry into and out of cells. But what's so clear is that this is not a mutation, it is an addition, it is an insertion. 12 extra nucleotides, 4 extra amino acids have been added, and this is clear by aligning the sequences with other viruses. This is the first and only discovery of a fear and cleavage site in a Sarbeco virus ever made.
See, he does the vertical alignment when you should be looking at the horizontal alignment, and it's so much more obvious. It's even bigger than that. I want to show you the real one while we're doing it, while he's doing it. Where is it? Here it is, down here. This is even a better alignment. Can you spot the difference? This is RATG13 at the top, and this is SARS-2 at the bottom. That's basically 500 amino acids, and there it is, PRRAR. It's a 100% match, except for that little insert. So by offering up RATG13, Shi Zhengli made SARS-2 look engineered by someone somewhere.
Out of many hundreds. It's quite striking. And you'd think that it would indeed alarm and interest everybody who saw it. But when Shi Zhengli published the sequence in Nature, she didn't mention it. She doesn't use the words furin or furin cleavage site in the paper at all. And her diagram of the first half of the spike gene stops just before it would appear. As Alina Chan has put it, this is like describing a unicorn and not mentioning the horn.
So that was their little conspiracy that set them off on this weird little tangent for their book writing exercise, I guess. And I got to explain why. That's just so boring, actually. It's getting kind of old. Here it is. He's doing it here. This is after Diffuse is leaked, by the way. He's still trying to keep the attention on Shi Zhengli. So this is Flotabar, a natural origin scientist, along with someone else whose name I can't remember. But guess what? Shi Zhengli's software is so old. It's MS-DOS based, and it always stops at the border, the S1S2 border. This is what they're talking about here. It should have kept going, but it always stops here. In 2000, what is this? I don't know, probably 14. It stopped at the S1S2 border. It stopped here, 2013 at the S1S2 border. She always, her software is so old, it always stopped at that same spot. She doesn't even care about the fear and cleavage site, because she's not even interested in that kind of stuff. She's only interested in the S1, not the S2. So the full threads here. I'll have to upload, I guess, these on the site. But it's Shi Zhengli's software is so old she can't even see the SARS-2 fear and cleavage site. That's how pathetic this whole conspiracy is.
That diagram stops there. But the end of the S1 section of the spike gene is there. Why not go on to the S1-S2 boundary?
You see what that did? It led to the whole weird conspiracy that Shi Zhengli has something to hide. Again, I keep harping on this, but when she published RATG13, she exposed the fear and cleavage site.
As you can see, the
She's not hiding it. She exposed it. Two arginine residues in it. That's her software so old she didn't even know what to look for. Which makes it more funny.
For by CGG. Well, that's unusual, because these kind of viruses do not use CGG for arginine very often at all. It's the rarest codon to use, and yet here are two of them next to each other, and this is the only occurrence of two CGG codons next to each other anywhere in any sub-ecovirus genome that we've found so far. No wonder David Baltimore, when he first saw this, said this could be the smoking gun. He later wrote back from that under pressure from colleagues, but that's what his first thought was.
CGG would be used for like a mammalian vaccine. So it's not special for humans, it's just special for bats, or mammals in general, bats in particular. So it would be something Baric would use if he wanted to infect bats, CGG. It's also easier to grow mammalian cells. So you're doing all kinds of tricks like Baric could.
Well, maybe Shi Zhengli wasn't interested in furin cleavage. Maybe that wasn't her thing. I'm afraid it was. She'd spent a lot of time, as had Ralph Baric, in the months and years leading up to the pandemic.
This is not really valid, by the way. He's kind of got some hodgepodge stuff here. Neither one of these are bat coronaviruses. This one's a rat one, and this one's a protein. And neither one of them are done at the Wuhan Institute of Virology. These people aren't at the Wuhan Institute of Virology. I believe they're. I know this one isn't. I believe these aren't. I know this one's in Shanghai. This one, I don't know where it's at. I don't think it's in Wuhan. But even if it is, it's a rat virus, not a bat virus.
Interested in how much furin cleavage
there's zero interest by any Chinese scientist before the pandemic.
A limiting step in how well viruses
furin cleavage
infected. Here is a paper that she published in 2017 on a rodent virus, where she draws attention to a very similar furin cleavage site. And here's a paper published by her colleague Shi Zhengli, in which he inserts a furin cleavage site into a gene, albeit not a
viral one. You see, he's so smooth, he lies, and you can't even tell he's lying. This is not even a virus. It's a protein.
Gene, but an antibody gene. A 12-nucleotide sequence. And here's a virus that they do mention in their Nature paper, a cat virus, coronavirus, which has exactly the same furin cleavage site. So they would know that the sequence PRRAR would create a furin cleavage site if inserted. Now, as I say, the news of the furin cleavage site alarmed many Western scientists when they first saw it, and that letter from Christian Andersen to Anthony Fauci resulted in a flurry of activity. For a while, we weren't able to read the emails, but then we did. And this is what Fauci said to Farah. I just got off the phone with Christian Andersen. He related to me his concern about the furin site mutation in the spike protein of the currently circulating virus. I told him as soon as possible, he and Eddie Holmes get a group together, examine the data, and if necessary, report it to the FBI and MI5. Quite right, too. That's the right response. And indeed, later that day, a meeting was convened between Fauci, Farah, Collins, Valence in the UK, and a bunch of virologists, including Andersen and others.
He's again lying by omission. He does not bring up the biggest heel turn of them all. Instead of Fauci calling the FBI, he got off the phone at midnight, now Saturday morning after being told it's engineered, and he picked up the phone and called Ralph Baric. Tony Fauci invited Ralph Baric to the February 1st call. Christian Andersen and Eddie Holmes are on record that they did not want him there because they knew he was implicated with this kind of research with the Wuhan Institute of Virology. But Tony Fauci invited him on there, and not even Farrar knew it. No one knew it. I got Eddie Holmes, not Eddie Holmes. I got Christian Andersen to admit to me he had no idea Baric was on the February 1st call. That changes everything because everyone thought, myself included, that he wasn't on it. And it was always weird. What on earth made these guys go from 100% it's engineered to, maybe it's not? Well, I guess we'll get into that later. But yeah, that is the big twist that happened in the past couple of years. Ralph Baric gave that up during his 2024 testimony. He thought it was public record. It wasn't. That's a big deal. I think that's Ralph's way of basically saying, if I go, Fauci goes with me. But yeah, of course, he's not going to bring that up because it doesn't fit his narrative. This is the way the guy organizes his life, through a narrative. And if it doesn't fit his narrative, it's not evidence to even be entertained. So, lesson for the weary: when you get that old, get off stage.
And the upshot of that meeting was that they agreed that they would write something.
When you no longer have hair, I don't think people want to hear what you have to say. How about that?
Something about this, and that Andersen would be tasked with drafting it. And after the meeting, Farah and Andersen and Holmes all still thought it was quite likely, as a hypothesis, that the furin cleavage site had been inserted. But sometime over the next few days, they changed the line.
That is borderline criminal what Fauci did to them. They did not want Baric on the call. They wanted to discuss it without him, and Fauci invited him in.
They were going to take in this article they were drafting, and eventually ended up writing something that...
Fauci can't even spell furin. That's interesting. He doesn't even know what it is. Furin site mutation. Yeah, I really want to be clear here that I would imagine that in the USA, Fauci is being a little vague here in this email. Christian Andersen read it, and the way he talked about it is that Fauci would call the FBI. But if you read here, Fauci's being a bit slick. He'll say, well, why don't you guys call the FBI? But Fauci never called the FBI. He's on record as saying, I never did. So again, Fauci doesn't call the FBI. He called Ralph Baric. After being told it's engineered on Friday night, January 31st of 2020. The first guy he calls isn't the FBI or the CIA. It's Ralph Baric, the guy that proposed to insert the fear and cleavage site in 2018 and tested on Chinese bats in Wuhan.
Ruled out a lab.
I mean, obviously, Fauci and Baric and I believe Munster have been talking offline privately before this happened. They're like, shit, that's ours. Let's hope this doesn't blow up the world. So it was a brilliant move by Fauci. It was just brilliant.
Operation.
I admire the guy. He knows who I am, by the way. I've asked him for an alibi. I mean, why not?
Altogether. What had changed? Christian Andersen is on the record as saying that he'd had a good look at RATG 13 for the first time, and this convinced him it was different. But that's not really plausible because he had been tweeting about RATG 13 a week before. True, by the end of...
That's interesting. I didn't know he said that. So even Christian Andersen had a backdate when he knew about RATG 13. So he's admitted in a way. I'm not getting into this.
That week, there was indeed a possible excuse for why they could start to rule out a lab leak, and that was the news from Jeremy Farrar to Francis Collins.
In a way, he just admitted that Christian Andersen needed RATG 13 to see that it was engineered.
That reports were coming out of a 99% similar virus in a pangolin, the missing link. And Collins quite rightly replied, very interesting, does it have the fear and cleavage site? And the answer was no, it does not. And nor is it 99% similar. Once we saw the whole sequence, it's only 90% similar. It's not the missing link. I won't go into the pangolins in any detail because they're a red herring, but it's important to note that the effect of this announcement was to throw a lot of us off the scent for a while. Whether that was its purpose, I can't say. But before he published his paper ruling out a lab leak, Christian Andersen knew that the pangolins didn't help. Unfortunately, pangolins don't help clarify the story. The paper appeared in March.
If you want to replace the pangolins with North American white-tailed deer, a lot more will make sense.
March of 2020, pre-printed in February, the proximal origin of SARS-CoV-2.
So he's completely eliminated. I don't know if he brings it up. I'll bring it up here. So on February 1st, they do the teleconference, and Ralph Baric has secretly listened in quietly without announcing his presence to their evidence, and he now sees what it is, and it's pretty damn good. He's got RATG 13 to show this thing has got a fear and cleavage site in the middle of it. And so Ralph Baric waited 2 days and ambushed Andersen on February 3rd of 2020 during a bigger meeting, the National Academy of Science meeting, where Fauci organized that with the FBI and the CIA and all the bigwigs from God knows where, the White House and whatnot. And in front of everyone, Ralph Baric ambushed Andersen. Attacked was the word that Christian Andersen used. Ralph Baric just attacked me for suggesting this thing is engineered. I wonder if he himself is implicated too. So he will not bring this up because it doesn't fit his narrative. But it was Ralph Baric who basically attacked Andersen and gaslit him into the idea this thing is natural. And if you're Christian Andersen, you're not a coronavirus specialist like Baric is, you kowtow. But give Christian Andersen credit. He tried to, he wanted to call the CIA and FBI, and he got ambushed by Baric. It's the story inside the story. Baric has attacked, ambushed Christian Andersen and gaslit him out of this idea that this thing's engineered. So when that came out, I thought we would all be like, we as lab leakers would be like, oh, well, this is pretty obvious now what happened, isn't it? Baric did it. And he just basically drove everything. He drove the whole cover up. He basically is the ghost author behind the proximal origin paper. Because they were going to, do something completely different up until Baric attacked Christian Andersen. So he doesn't bring that up. And why not? This is I'm not going to say it. This is CCP level, propaganda, what he's engaged in here. He's just an old man who's very prideful, like any British bitch.
And it said, we do not believe
he's going to go down with the boat on this one.
And in a press release from Scripps, Andersen went further and said it rules out laboratory manipulation. Now, I was misled by this paper. This was what led me to go and say to colleagues who were asking about this in Parliament in the UK, no, it's been ruled out. It wasn't a lab leak. There's good evidence it's been ruled out. But I hadn't read the paper very carefully. When I did, I found the arguments rather flimsy. I mean, the main argument was that the fit of the receptor binding domain to the human ACE2 receptor was good but not perfect. And if people had done it, they'd have made it perfect. I'm not sure that's a very rational argument. But it was only many, many months later that we found out that Christian Andersen had not changed his mind after all. He said on the 17th of March, we do not believe any type of laboratory-based scenario is plausible. On the 1st of February, he thought it was frigging likely. On the 2nd of February, after the meeting, he still thought natural or escape were both options. And on the 16th of April, a month after publishing the paper...
So, you see what happened here. On February 3rd, Christian Andersen has been attacked by Baric. And, he's been gaslit. So, it's what happens. So, you kind of see him. He went from pretty convinced it's likely to, well, I don't know. It's just what happens. If someone older, wiser than you attacks you, you kind of kowtow. It's just the way humans are wired.
He's still not fully convinced, can't fully rule it out, and thinks the fear inside could still have been inserted. Now, I'm sorry, but where I come from, publishing a scientific paper that says the exact opposite of what you think in private is not good scientific practice. But those of us who have written to the editor of Nature Medicine requesting that this article be retracted or corrected have not even had the courtesy of a reply. We do have quite a good explanation of what had caused them to change their line, if not their mind. And it comes in an exchange between Andrew Rambo and Christian Andersen. Given the shit show that would happen if anyone serious accused the Chinese of even accidental release, my feeling is we should say that given there's no evidence of a specifically engineered virus, we cannot possibly distinguish between natural evolution and escape, so we are content with ascribing it to natural processes. And Andersen replies, yep, I totally agree, that's a very reasonable conclusion, although I hate when politics is injected into science, it's impossible not to. Now, you can't read that exchange and come to any other conclusion that the decision to write the paper the way they did was political rather than scientific. In September 2021...
He just completely, missed one of the greatest things ever. We're in two different planets, basically, at this point. I gotta think about a search for that. When I came across it, on February 3rd, the next day, this came up. I'm not gonna get into how it showed up, but basically, one of the congressional committee guys brilliantly brought it up during testimony and got it into written record. I should mention, this is on February 3rd. I should mention that Ralph Baric pretty much attacked me on the call at NASM, essentially calling anything related to the potential lab escape ludicrous crackpot theories. I wonder if he himself is worried about this too. I'm just trying to get your side on this. Can you read that again? Ralph Baric was just completely caught off by that, and he didn't have a good answer. He came up with the BSAG theory, but that fell apart because he never mentioned this, the fear and cleavage side of that meeting either with the CIA. So this happened on February 3rd. That is a holy shit moment. When I read that in 2024, I was like, holy shit, that explains so much. I was just like, wow. I don't think Ridley reads anymore.
A document was leaked to the press that should have been made available from the very
those
start.
Those that like to talk don't like to read, and vice versa. I'd rather be reading than talking, but here we are.
Proposal. Lots of people knew about its existence and chose not to tell us about it. It's a very important document.
It's just, here we go. He's now cropped out the University of North Carolina, which is down here. Well, there it is. Never mind. It's right there. At least you got that.
There's an application from Peter Daszak on behalf of the EcoHealth Alliance, Wuhan Institute of Virology, and a number of other groups for $14 million from DARPA to investigate defusing the threat of bat-borne coronaviruses. And it stated, halfway through the text, that they were interested in furin cleavage sites, and that they were interested...
They is Ralph Baric. It is, in testimony, it was my idea and all my idea.
They couldn't find one in putting one into a...
You see how he goes collective when he doesn't want to incriminate Baric, when he goes singular when he wants to incriminate Shi. It's just... This lab leak debate's turned into my social thought experiment. And it's no longer a biological debate for me. I'm really having fun, though, with this social thought experiment. And I think if there's a follow-up book to be made or done or thought about or write about, it's the way humans can lie to themselves and dig into their preconceived notions. And I think it's theological. I don't think it's biological. I don't think it's psychological. I think it's theological. This need to be a part of something, this group, this movement, is very strong, and it's a theological one. And he claims to be an atheist. I make an argument he's not. He's a bit of a quack, a creationist, if you will. But I'm not getting that here. But I think it's theological for him. He needs to be a part of something. He wants to be in front of the crowd. He wants them nodding with him, listening to him, like a preacher, like a sermon. He's very theological when you listen to him talk. Self-righteous indignation kind of thing. He's got a theological bone inside of him. That's for sure. I don't get too weird here, but they, again, is Ralph Baric. That is me. Ralph Baric's human airway epithelial cells, potential fear and cleavage site. This is all, we is I, Ralph Baric, Get that percent. This is all Baric's, stuff. This whole paragraph is written by Baric. So, and it was written in English. And it's obviously, when it came out, I was like, wow. That looks like Baric's writing, because I actually kind of like the way he writes, because it's kind of like to the point. It's not this fluffy words like he would use. It's just, we will do this. We will insert a fear and cleavage site. Hell yeah. Get right down to the point. Get the meat and bones. That's kind of how I enjoyed it. I can't read Shi Zhengli's writing because it's terrible. her English is, better than my Chinese, but it's written in English by Ralph Baric. And, he's the only one that could do it because he's the one that wanted to do it. I don't get into why, read the book. It's like chapter 14 where I get into how it's biologically impossible for Shi Zhengli to do it. Vero cells mainly are, Vero cells delete the fear and cleavage site, just without getting too far in the weeds here. And that's only Shi Zhengli had Vero cells, and really Baric only had human airway epithelial cells. You need human airway epithelial cells. It's basically like a human cadaver that's been donated to the UNC lab. That's basically what happens if you're a lung donor on your driver's license. You wind up reincarnated as a petri dish in UNC for Ralph Baric's experimentation. But
we will introduce appropriate human-specific cleavage sites and evaluate growth potential in monkey cells and human airway epithelial cultures.
No HAE in the WIB.
Now, some people have said, oh no, they were talking about putting it in at the S2 site, not at the S1, S2 junction. No, the diagram makes quite clear that they're interested in both sites.
That's Baric's diagram, literally figure C. It's funny, going back to the theology side, people use the collective when they don't want to assign blame, and they use the singular when they do.
Earlier draft of the document, we were even more shocked because we found.
He's going into the back. He's going back to his collective tongue. So this is interesting, but let him.
Peter Daszak reassuring Shi Zhengli in a marginal note that although he says the work will be done in North Carolina, a lot of these assays can be done in Wuhan.
Essays are a PCR test, not fear and cleavage site work.
And the reason for that is because the biosafety level 2 nature of the work on SARS-CoV in Wuhan makes our system highly cost-effective. Much cheaper to do it in Wuhan. And Ralph Baric replies, China might be growing these viruses under BSL-2. US researchers will likely freak out.
What he's talking about is a comparison really to Ebola, Marburg, and whatnot. At least Peter Daszak was in BSL-4. So, in other words, that was Peter Daszak trying to say we're cheaper than the competition. The competition was bidding on Ebola and Hydra and Nipah. The competition was people like Vincent Munster, ironically.
Did they freak out? Here they are, freaking out. Anthony Fauci, serial passage in ACE2 transgenic.
Yeah, if you think a virus that floats 60 feet in air leak from a BSL-2, I don't think your head's screwed on.
Nice.
Anyways.
Jeremy Farrar, exactly. Francis Collins, surely that wouldn't be done in a BSL-2 lab. Jeremy Farrar, Wild West. Let me remind you of the timeline again, of all the gin joints in all the towns in all the world. This virus walks into the one city where this kind of research is happening the year after someone has proposed to put a fear and cleavage site into a
That's someone's Baric.
Zika virus for the very first time. That's quite a coincidence. Now, a few loose ends, and then I'll wrap up. I'm sorry I've gone on so long. There's a mutation in the human version of the virus that happened very early.
D614G. Watch this. D613G. Where is it? Ralph Baric studied D613G in SARS-1 in 2006. That's pretty trippy. If you figure that out, let me know. But I'll just leave it at that.
Early in the pandemic, it stabilizes fear and cleavage. It's a response to fear and cleavage. It implies that the fear and cleavage site was new, had not been in that virus forever. Chinese government banned the sale of ex-laboratory animals in markets during 2020. 3 key lab workers, according to US intelligence, I can't verify whether this is true or not, fell ill. They included Ben Hu, who was in charge of these chimeric viruses.
So this is a bit hokey. I'll be honest. I'm not going to try and poke a hole in it here. What did I do? Gordon. I don't know if I have his name there or where. There it is. I do have it. So that intel came from this guy, the weapons of mass destruction guy, and it was fed to him by a guy named David Asher, who got it from a walk-in. I think I should stick here with this real quick. David Asher was the one that started that rumor, and he got it from a walk-in. Here it is in my thread.
The way I figured out what the U.S. was doing was by investigating the Chinese. I kept running into U.S. grants and research and U.S. researchers in bed.
This is where the 3-6 started. This State Department dude. I've interacted with him a couple of times on Twitter personally, and I'll say it. I did that interview 2023 with Frank on my self-spreading vaccine theory, and he great job, Jim, for bringing in Lympho Wong. I'm like, yeah, good job for carrying Fauci's water. And I don't think we ever talked after that. I think he's I think he's Fauci's little, bag boy, basically. They think they're getting Fauci and China. I mean, I'm like, no, all you're doing is just covering up for Fauci.
With the Chinese lab
with
that
this,
we were
this
concerned
stuff.
had leaked
I mean,
this
he's
thing,
just a China hog.
developed it and leaked it and was funded by the People's Liberation Army to develop it, not to release it. We had no knowledge that I ever saw that implied deliberate. We did see things that I can't talk about. I did have lots of interviews with foreigners who had worked in Wuhan with the people in that lab, were in direct email and signal and other contact with those people, even as this was occurring. So I went and did, we knew it state. It's not the IC. We sent out cables to our posts and we asked, call in all the scientists that you can find each one of your posts in your country where you're located and who had worked in Wuhan and find out what they know. Well, we found out an awful lot just based on that. And what we found out is that there were people in that lab who apparently, not only fell sick in November, and this came from State Department non-classified information. Almost all of our cables related to this topic were sensitive but unclassified or unclassified. I mean, we did have
See, it changes from November to
some
October.
classified cables.
20-year-old postdocs don't get sick with COVID.
And we did have some people that will say, people can walk into a U.S. embassy and ask to talk to the State Department. They'll really, if they don't want to talk to an intelligence agency, they're going to talk. They have to ask to do that. They want to talk to state will talk to them. And so we collect a lot of independent corroborating information that, frankly, a series of accidents and mishaps occurred involving the most dangerous, convolutional, developed dual-use capability funded by the People's Liberation Army in that lab. And one of the first, maybe the first cluster of people that fall sick that we knew about was in the Wuhan Institute itself, and they were people who apparently were working on COVID-19. And that doesn't come out of the U.S. government fact sheet. That comes out of an interview I did with 2 foreign government scientists who were working there.
There, he just said it, where it came from. He interviewed some random dude online, and he claimed to be working there that heard something about someone.
In that time period. And they were
online rumor.
They told me what they believed had happened, and I asked how did they find out? Well, I'm not going to say who they talked to in the staff of the Wuhan Institute, but they certainly talked to them.
Where does all
So that's where the whole 3-6 started, with this rumor started by this guy right there. So, yeah, I'm not getting into that here. Let's just stick to this. But, yeah, it's not even a good piece of evidence if you're sick or not. You know, it's really a good piece of evidence is biological proof, in vivo proof, in vitro proof. We have that at Rocky Mountain Lab. We have that at UNC.
Experiments, and Ping Yu, who, as we saw, was the person who described lineage 4.
Again, if you think that Shi Zhengli, Bin Hu, Peng Yu, or one of our underlings is sort of that fear and cleavage site you published RATG 30 exposing the fear and cleavage site, I think you're a simpleton. Oh, I know you are. You can think whatever you want.
The man who developed the COVID vaccine very early in the pandemic in China.
This is brilliant. This is brilliant. Zhu Yusin is based in Beijing. So if he fell from a rooftop, which is another bizarre rumor, but whatever, let's say he did, it would have been in Beijing, not in Wuhan. So the guy literally has no connection whatsoever with the Wuhan Institute of Virology. He has no connection with Wuhan. Maybe he's on a paper with them, which I think he actually was. But just because he co-published with them doesn't mean he lives with them or lives in the same city with them. So he lives and works in Beijing in a Beijing lab. So and if he did a patent basically of a vaccine, he started developing it after Moderna and after Baric and after others. You know, no quicker than them whatsoever. It's just one of these bizarre conspiracies. You know, they found some dead dude in Beijing. That is his evidence of a lab leak. A dead dude in Beijing. He got up there. This biology PhD reporter, writer, book writer, wants to get up there and talk for an hour. I've been talking for an hour, and he has nothing. At least we have something.
Falling off a roof.
To
summarize, ladies and gentlemen, in 2017-18, in Wuhan and nowhere else.
This is it.
This is it.
Shifted their priority to 20% divergent viruses. Baric shifted their priorities to 20%. Real quick. This guy is so smooth. So smooth. It's just unreal. Yeah, Baric, where is it? Which one is it? 20%. This is RB is Ralph Baric has already generated SARS-like chimeras, 20% different than SARS-1. Well, guess what? SARS-2 is 20% different across the whole genome. So this is Ralph Baric has done this. Not the WIV. It literally says RB has generated, not the WIV. So
yeah. She shifted their priority to 20% divergent viruses from SARS, sequenced at least one.
I think he just reads what I write and then just kind of pigeonholes it into his own little theory, because they all do. There's a bunch of closet readers that read what I write, I've come to learn, and they're just chicken shit cowards. They're going to hide in the closet.
Fun lineage 4 Sarbeca proposed to insert a fearing.
There it is. See, lineage 4. I can't even keep up with all this stuff. I thought I had it here. Let me get rid of that. Sorry, this is dragging on too long, but this is kind of getting fun now. There, I'm going to come back to that. Don't let me forget about that. Where you see did it again. Where is it? Lineage 4. Here it is. Lineage 4. So if you Google AI search lineage 4, I'm not sure where it comes from. Neither do I. It's an African coronavirus. It's not even a Chinese coronavirus. It's lineage 4 was in the Yu Ping thesis. We don't even know what her name is. It was Yu Ping or Ping Yu, and it was Yu Ping in the sick report. It was Peng Yu and on the papers. He can't even get it straight. So lineage 4. That's right. She was allegedly one of the sick. I forgot about her. But yeah, no one calls it lineage 4. SARS-2 is lineage 2. To be precise, it's clade 1b. I've gotten this debate with Saga recently. So it's a lineage 2, clade 1b. Everyone considers it lineage 2 except for Ridley, because it's part of his little conspiracy theory of this thesis that basically has been cooked up. So, and then here you see the way this whole thing spreads. So it's just, oh well, Ridley's smart. He has a British accent. He has written books. Let's follow him. This is where it came from. And Ridley's following some 20-year-old postdoc who doesn't even know how to classify RATG 13. It's a lineage 2, clade 1b. But he got up there and just did it right there. He calls it a lineage 4. And then, if you don't listen to me, which I don't blame you, here he is, Christian Anderson, saying the same thing. It's a conspiracy. The whole thing's just bizarre. It's just, you can't even get the science right. It's a lineage 2. Proposed to insert the fear and cleavage site. Again, that's Ralph Baric.
For the first time. Proposed infecting human cells and humanized mice with such viruses at a low biosafety level. And following years in Wuhan, a lineage 4 Sarbeca virus appeared.
He doubles down on his lineage 4 conspiracy. It's lineage 2.
A newly acquired fear and cleavage site in the very same city, leaving no trace in any animal in the markets or anywhere else.
North American white-tailed deer, American mink, American lab bats called the Egyptian fruit bat, American deer mice, and Syrian golden hamsters. You see, he won't even mention the hamsters. If he was smart, he would switch to hamsters and not humanized mice. But he's not smart.
And the Wuhan Institute of Virology misled the world, whether intentionally or not, about the name and date of sequencing of certain relevant viruses and took their database offline.
Now, if you think that database went offline when Shi Zhengli published RATG 13 on January 24, 2020, you probably are as dumb as Ridley. So I don't get too mean here, but I mean, he's too fun to pick on.
Now, I'm often asked, if you think all this is important, why haven't you published it in the peer-reviewed literature? Believe me, we've tried. We've submitted papers saying all of this again and again.
So real quick, I'll just pimp my own little thing here. We have, myself and Jeff Sachs, have published peer-reviewed proof of a Wuhan lab leak. I'm not going to get into that here. But, you know, I at the very end, I drive it home. I love my closing line here. I thought it was my own pietà. In the end, peer-reviewed literature revealed a bitter truth. Fauci, Baric, and Munster concealed evidence of a likely U.S. lab origin of SARS-CoV-2. So that is the peer-reviewed proof is all of this right here. You know, it's nearly 100 plus citations. So, and it's mainly the in vivo models, the transmission models that someone like Vincent Munster used to aerosolize the agent. That's where I came from. People always ask about my background, and I was kind of being a bit, you know, dark about it because I don't want to talk about it, but I talk about it a little more now. But yeah, I was, you know, essentially viscosity specialist. And I understand when you study viscosities, you understand aerosols. And when you understand aerosols, you understand transmission. When you understand transmission, you'll understand the origin of SARS-CoV-2. So
they're
that's
always turned down by gatekeeping referees who want to be able to say, you haven't published this in the peer review.
This is the problem I'm having. Biologists want to look at the genome, and I just wanted to look at the in vivo transmission models. And I backdoored this thing, hence the name of the blog, Reverse Engineering. I knew Vincent Munster aerosolized the agent in 2022, and then I approached Ralph Baric in, I think, early 2023, and I said, look, you give me Vincent Munster, and I'll leave you out of this. And I halfway expected him to do it, because I was like, wow, if someone came up to me and said, I got your accomplice, and I'll just throw him under the bus and leave you out of this, I would basically help out. You know, I'm sorry, Mr. or Mrs. Accomplice, but I'm done with you, and you can go die for me, and I'll live. But Ralph Baric dug in on me. I couldn't believe it. So I received published something in Brownstone and dig them out. But in Dutch, whatever. I'm not going to do that here.
That's a circular argument.
So, yeah.
And on that note, ladies and gentlemen, I'll end.
We do have peer-reviewed proof of a lab leak. So this is interesting. He has listed David Asher, the guy that started the whole online rumor. Billy Boss isn't I like. A lot of these people I like. Bertel did a lot of hard work, but he kind of did it in the wrong way. But I'm not getting to that here. Alina Chan, I respect. Yuri, Gilles Demonov, I like. Olive Drastic, I like. You know, I needed them more than they needed me. All these people are great to me in the grand scheme of things. But there's one name missing here, and it's Major Joseph Murphy, the guy that leaked DARPA Diffuse and leaked it to Drastic in such a smooth way they didn't even know he gave it to him until his own letter leaked. So I'm not getting that here because I think I've talked about it in other videos. But just it's very telling that he doesn't even mention the guy that leaked DARPA Diffuse, who found it in a top secret folder written by Ralph Baric, saying, I will insert the fear and cleavage site and test it on live bats in Wuhan. So it just tells you where he's at. It's online, on Twitter, all day, every day. And that's all he thinks. That's his worldview, Twitter. And these people are all on Twitter, twittering about lab leak. And that's basically what's happened here. They formed some little bubble, basically, around themselves. That they're right and you're wrong. And, you know, this guy in particular. But, yeah, it's a bubble. You know, this is all this is. It's basically proof that they're all, you know, just in this self-interested bubble. It's almost worse than the virologist. I have an easier time communicating with the virologist than I do lab leakers. So, and none of them and all of them have been given an opportunity to answer the simple question: why did Shi Zhengli of the Wuhan Institute of Virology publish RATG 13 exposing Ralph Baric's fear and cleavage site? And they can't answer it, but I can.
I would just like to acknowledge the many, many people, this is not an exhaustive list, who have contributed to this knowledge. Most of it didn't come from me, it came from these people. Thank you very much.
Very religious, isn't it? You know, the claps, the applause, even the whole, you know, the stage and the setup. You know, the Turin, you know, cloth in the background. I won't go that far in my whole analogy here, but it's got a theological sense to it, science, and most of the biologists I come across have a biological need to be included, be a part of something.
Thank you for.
They're looking for a purpose in life, and I think they find it in science. So I'll leave it there. You know, they do a Q&A. This thing's on YouTube. You've heard enough of me. I'll upload this on my own little channel just to get my own little perspective out. You got any questions? Let me know. Again. Reverse engineering the origin of SARS-CoV-2. You know, maybe one day we'll get it out, but even if we don't, who cares? I can still call it the 1,000-year project. Just document, and you know, some future historian can look back when we're all dead and say, yeah, I think the guy was onto something. But I'll leave it there. Good night. See you next time. Bye.